morning, everybody. Thank you very much for the opportunity for me to share my research. I would like to thank firstly to Professor Joel, Professor Franco, Professor Mendoza, and also Professor Raimundo, and also Professor Angela, is the moderator this morning. Really appreciate for the invitation and opportunity. This morning, I would like to share about the biofilm and also the anti-biofilm. How is natural anti-biofilm need to be developed to control, and one of the alternative strategy also to combat the bacterial biofilm everywhere. As we know, biofilm is assemblage of surface associated with microbial cell, and also some can be also eukaryotic cell and become biofouling. And this structure usually enclosed with extracellular polymatrix substance like protein, and then also lipid and extracellular DNA, and also polysaccharide. Majority are that compound. And we can find also biofilm is everywhere, like Professor Joel mentioned this morning also. So we can find everywhere in near with surrounding our area, in the house, in the office, and also including in our body, like our teeth, dental, and then also in the lung, and also in the gut. So actually this is everywhere, including in our food. So we need also need to require attention to this structure. Biofilm formation.
Professor Watorangi, I think you're on mute.
Okay. Yeah, so they can be free floating, and also then they start to attach to some surface or matrix. It depends on the situation, like maybe deprivation or starvation of nutrition, and then also stress in the environment. So they can start to attach and then form the weak attachment like van der Waals, but then it can start also to form the permanent using the pili flagell, for example, like this. So they start in the free floating phase, and then they start to attach like using pili flagell or other compound, and then start the other step. We will discuss this later for detail. So we start the problem. What is the problem related with biofilm actually? It is everywhere problem. We know also some are application of biofilm, but mostly are become problem for us. And let's see, this is like problem in the for related with human infection, for example, like cystic fibrosis. Related with Pseudomonas infection in the lung, and mostly they become resistant, actually specifically become resistant to antibiotic resistant, to treatment of antibiotic. So the treatment become a problem. The other problem also can be related with the water distribution system, especially if the system is the water can be used as a source of our drinking water. So this can be a source for infection, especially waterborne, foodborne or waterborne bacteria. They can be contaminated through this biofilm compound. And we can find also on various kind of food, and also in food industry, like everything the food on the all of the surface that contact with food is there is a possibility can be a source of infection of the bacteria. So many type of food, especially the one related with high nutrition and high fat or high protein, and also high moisture, and also high temperature, mostly related with the formation of the biofilm. As we see also in many type of industry, especially related with food industry, also there is a problem related with the formation of biofilm. The other thing, the most important thing in bacteria living in biofilm, usually they have very specific or different property compared with the free floating or planktonic bacteria. Because mostly in the biofilm, bacteria can share transfer the many kind of gene, including antibiotic resistant, including virulence related gene, or any kind of gene that can be transferred inside of the biofilm. And also they become very resistant with various treatment. So biofilm formation confer the many advantages, especially for the bacteria, microbial cell. For example, in food industry environment, because they can they become resistant to physical treatment, like including desiccation, and also like mechanical resistance, like liquid stream in the pipeline, and also chemical protection. Like usually in the industry, they use like many kind of disinfectant, but mostly bacteria in biofilm also become resistant to various type of the chemical treatment. And the other thing related with health field, also medical field, we know that they become resistant also to various type of antibiotic due to the physical structure of they have. The matrix extracellular, especially they protect the interior of the community of the bacteria. So what is actually the biofilm community? The antibiotic resistant appear to because of various strategy. For example, like one is because of slow or incomplete penetration of antibiotic due to the physical structure formed by the biofilm to protect the bacteria, and also alter chemical microenvironment within the biofilm, and also subpopulation of microorganisms in the biofilm. For example, like sometimes they can also form the spore formation. This is one of the book chapter that we write related with biofilm, the structure of biofilm in gram-positive and gram-negative. And we found that a lot of mostly specific bacteria, they have also specific compound of the biofilm. For example, like mostly they are constitute of polysaccharide, and then protein and lipid, and also extracellular DNA, but they can be different in they can be variation in the percentage of the compound, and also some also they have specific brands. Sorry. For example, this is in Vibrio cholerae, and then E. coli, and also Salmonella. They have majority, they have protein and polysaccharide and also DNA, but different in the percentage also, and also a variety in the specific kind of the sugar and the protein they have. So when we look about the step by step of the formation of the biofilm, it starts with the look at this figure. Start with the planktonic or free-floating bacteria. They swim, but then when the situation is changing, like in the environment or nutrition starvation, but then they start to attach to the surface of the matrix. It can be live or non-living matrix. So they start, and then they start to produce the extracellular compound, and then they start to produce the polysaccharide, and then protein and lipid and extracellular DNA to form the biofilm. And then the biofilm start to enter the maturation process. And after that, also, we know that this is the cycle, because when the situation, when the environment is changing, like nutrition, and then become high enough, and then they can be start to disperse. So it starts again the cycle. So as we know, this is the cycle. For each bacteria or for each species of the bacteria, majority the cycle is the same, but only the compound that they need to form the biofilm or also to disperse the biofilm, it can be different. So actually we need to treat for each species of bacteria, we need specific also natural, well, we need specific also antibiofilm to treat or to handle. So the important part of the biofilm formation step is the communication. As we know, communication is very important, not for human, for animal, but also for microbe, right? In a microbe, for example, like if they know each other the language, so they can communicate, and then they know that they have the same purpose, they have the same goal. But if the communication, if the language they cannot understand, then they cannot cooperate. So as we know that we call this process is quorum sensing. The quorum sensing, like for example in gram-negative, they use majority they use acyl homoserine lactone. So the acyl homoserine lactone produced in the cell in the lux family, and then they secrete to the extracellular environment, and then when the concentration is enough, reach the threshold, and then they go inside the cell, and then the gene target become expressed. And we know the gene related with the communication or related with quorum sensing is many kind of gene expression, especially like related with gene pigment production and also virulence associated gene. So many kind of gene, also the inducer of the gene expression related with quorum sensing, and including in biofilm formation. So as we know, after the communication, and then they start to form the biofilm. So we realize that the communication is also important part of the biofilm to start the formation, and also for maturation, and also to mature biofilm to be dispersed. So this is interesting part that can be used as a tool to control the biofilm formation, to inhibit for inhibition, or to destruct the biofilm. So one of our study is to develop many natural antibiofilm from microbe, from various sources of microbe. So as we see that this is one paper, they summarize like many natural antibiofilm can come from phytochemical and also biosurfactant, and also antimicrobial peptide. For the antimicrobial peptide, it can be mostly are to control or to downregulate the quorum sensing molecule, and also the modulate host immune response. So this is like the strategy related with antibiofilm or also inhibition in quorum sensing. For antibiofilm strategy, it can be using modification of the surface for antimicrobial coating to inhibit the adherence, and also modulating cell adhesion gene, and also inhibiting the matrix formation, and also inhibiting the quorum sensing mechanism. The quorum sensing mechanism inhibition, it can be used when the biofilm have not been started yet, the formation. It can be first step or also in the late step of the biofilm, because as we know, the quorum sensing is also very important for biofilm maturation and also dispersion. And so this is 3 steps of the quorum sensing inhibition or anti-biofilm. So beside that mechanism, there are many kind of approach also using bacteriophage therapy, or also combination therapy using anti-biofilm or combination with antibiotics, because mostly biofilm only control the biofilm, but mostly they did not inhibit or kill the bacteria. So some approach also they use antibiotic also, like combination therapy. So this is, we would like to share our study related with anti-biofilm. So actually there are 3 kind of approach that can be used. Like firstly is biofilm prevention, and then secondly is biofilm destruction, and also prevention of dispersion, because when the biofilm start to be dispersed, sometimes it can be contaminated, and then they also looking for other place to form another new biofilm. So for prevention of inhibition, it is important to stop or to inhibit the communication, or also to destruct the biofilm. We explore various kind of anti-biofilm from various resources of microorganisms, especially from the marine, and also bacteria from the phyllosphere, especially from the plant that we normally use as a salad, freshly salad vegetable. So this is one of our paper related with anti-biofilm to control food spoilage, biofilm forming bacteria. For inhibition of the biofilm, we use Chromobacterium violaceum as the indicator strain. As we know, this bacteria, they produce the pigment, violaceum pigment, and they use the acyl homoserine lactone to communicate and then to produce the violacein. So this as an indicator bacteria. And we validate, this is one of the some sample that we use. This is actinomycet. And then we validate the quorum sensing mechanism, anti-quorum sensing mechanism, or we can say quorum quenching. So this is the activity they have. And also we assay also for the inhibition of the biofilm using the actinomycet crude extract. And now we use also the supernatant for efficient application. And we treat for food spoilage bacteria like Bacillus cereus, and then Subtilis, and also Salmonella putrefaciens. And some of the sample, they show also quite high activity. And this is for destruction, also for same pathogen related with food spoilage. And then we do also the scanning electron microscopy. And this is the control, the biofilm formation of the Bacillus cereus. And then after treat with crude extract, we can see that the biofilm can be disrupted using this compound. And this is for Bacillus subtilis, using other sample for actinomycet. And during the electron microscope, also we analyze the EDS spectra to see the element before when the biofilm still form, and then after the treatment. And then we got that data that So especially related with carbon, it showed decrease after the treatment with actinomycet compound. It showed, and also for aluminium, also it decreased. So we can see aluminium, it's also important in the formation of a microcolony to attach to some specific matrix. But the other mineral also they show increasing after the treatment because of the disruption of the biofilm. And since the application we would like to use to control the biofilm related with food industry, with food related with food processing process. So we also check the stability of the compound for specific range of pH and also temperature to make sure that this compound is still active during the food processing step. And as we know that mostly instrument in the industry, also in food industry, is using either plastic or mostly are also stainless steel. So we see also the inhibition using the compound when the biofilm form in the stainless steel material. So we see that the activity, the compound also still active when the biofilm is formed in the matrix of stainless steel. And beside the related with food spoilage or foodborne pathogenic bacteria, we also interest to control the biofilm related with aquaculture industry. Especially for Vibrio harvey and also Aeromonas hydrophila. And this we check that the compound also increase the survival of the fish. This is tilapia fish. After we compare with antibiotic treatment and also with the anti-biofilm. The one infected with Aeromonas hydrophila. And this is the scanning electron microscopy after the treatment. This is intestine of the tilapia before when the biofilm, this is control negative, control positive control, and also after the treatment with anti-biofilm. And we see that there is disruption or destruct the biofilm in the intestine of the tilapia after the treatment using antibiofilm. And we check, since this compound we use for food spoilage as well as for the in aquaculture, related with aquaculture. And we check the bioactive compound. This is from the supernatant and also from the crude extract. What is the major compound of the the actinomycet extract that we have. So overall, we can see that anti-quorum sensing or anti-biofilm activity of the bioactive compound produced by actinomycetes and also philosphere bacteria. This is promising also to inhibit the biofilm formation and also destruct the biofilm formed by various pathogenic bacteria related with food or related with aquaculture. I would like to share a little bit about our ongoing research. We are also interested to study related with bacteriophage. We did research. This is a last year publication related with bacteriophage to control the enterotoxigenic E. coli. This is from various kind of food. Where they can control this pathogenic bacteria. And we also already studied the NGS for this phage that we did not find any antibiotic resistant gene or also virulence associated gene in the bacteriophage. So right now we are interested also using this phage to inhibit or to destruct the biofilm of the related with foodborne and food spoilage bacteria. We did also a biofilm assay using polystyrene because it's also material that normally used in the industry, including food industry, and also assay in the stainless steel coupon that we show that we have that data that it can be inhibit or destruct the biofilm in polystyrene or in stainless steel for the bacteria related with foodborne. And then we found that the occurrence of many biofilm-based pathogens and their multiple antimicrobial resistance. Of course, it can be a major concern in many areas, including related with food, with health, and also in aquaculture. So this is important, the exploration of bioactive compound of natural antibiofilm agent from various sources. This can be used as an alternative way to combat the bacterial biofilm. Thank you for your attention. I give to the moderator.